I was initially drawn to the Estonian Biobank thanks to the great vision of the founder of the biobank, Professor Andres Metspalu, who's really been a visionary in Estonia in creating such a huge population-based biobank. I think that there are many elements of the Estonian Biobank that makes it unique and that makes it an attractive place for scientists because we have extremely rich data, both genomics and metabolomics, but also very detailed electronic health records covering primary care, secondary care, and all health records in Estonia are brought into one central e-health database, allowing us to explore anything from medication use to diagnosis and procedures and combine that with genomics. And the other unique part is that we actually can go back to the individuals and we can return results. So it's a two-way street where we can do amazing research and we can also return results to biobank participants that is of benefit for them. I think such a project can only be done as a huge team effort, given that we need to have very different expertise brought together. And we need different skills, we need different points of view, we need different approaches, and we need to just find ways to work together to address difficult questions. And we also want people who have a big vision, who want to dream big, and who want to solve really difficult problems. So that definitely helps us forward, and everyone's contribution is important. And bringing people together with different views can be challenging, but that's also a joy in seeing the result in the end. So I think this is the main things that I've learned in the process. I think trailblazing in science is definitely working with the best in the field and also working hard. So we try to bring our best expertise to the table, our unique edge, whatever we have to offer, and learn from each other, because there's a lot of really smart people in different countries, and we've learned to collaborate on difficult topics. And there are many topics in genomics that we really cannot address on our own. We really need to work together and bring different data sets together, different expertise together. We need really strong bioinformaticians to work through these different layers of data. And I think collaboration is key, and working with smart people from around the world. I think Estonia is a really tech-savvy country and innovative, and people have kind of grown up with the mindset of we want to do big things, we want to do unique things. So this is kind of built into our DNA, or let's say epigenome. So we are really dreaming big, and we want to be among the best. We want to work with the best, and we want to do unique things. We want to be first, and we really strive to get there. I think being first means that you have to imagine that we can do something that some others might think is impossible, and also push ourselves to work hard, to dream big, and imagine that we can do it, and then ask for the necessary resources and believe that we'll get them. Then, once we get it, put the team together and start working towards the common vision, and really infusing everyone with this vision and feeling that we are first and we're working hard, and it's going to be hard because you're first. Just to figure out how we can do it at our best possible skill set and experience, and how we can build and learn from each other. In order to be first, you first need to have the vision, and then bring the people together who believe in the vision and want to contribute to accomplishing this big project or being first in a specific area. It will not be easy, but we are willing to work hard to pave the way and move forward in this direction. I think it's very important not to take no for an answer, and there are no obstacles that we can't overcome. We work together, we have a vision, and we ask for the resources that we need, and then we can do it. I think the fact that Estonia has gone through a difficult past and built up many things from scratch has created a kind of resilience and determination that we want to do it well, we want to be the best, and we can handle difficulties. So under difficult situations, we are resilient, we are determined to achieve our goal. I think once you have worked with long-read DNA sequencing, you cannot live without it. But that also on its own requires really strong bioinformaticians. So you need two things: long reads and smart bioinformaticians. I think the partnership with PacBio has made the transition from short read sequencing to long read sequencing a lot easier than I thought it would be. We have had amazing support in the lab, on the data analysis side, and excellent support from the bioinformatics. And this goes hand in hand with the more difficult part, that actually analyzing the kind of variants that we haven't worked with before because we haven't had long reads. So structural variants, really complex regions of the genomes that we haven't been able to analyze before, we don't have any experience with. So getting this kind of bioinformatics support from PacBio has been instrumental in our project. So for the 10,000 genomes project, we knew that we will have a challenge with genes that are very important in pharmacogenetics, so for drug response. And we had been looking into testing some long-read sequencing platforms, and PacBio was one of them. So once we actually did the comparison and saw what kind of data we got out of long-read sequencing, and then just the quality of the data that we got from the PacBio sequencing, we were convinced that this is what we want to use for our 10,000 genomes, and the question left was only whether we would afford it or not. Although pharmacogenetics was one area where we saw great value in the long reads and the quality of the data that we got from the PacBio sequencing, there are many other complex regions in the genome that we are interested in and would like to capture, because these are extremely important in the clinical setting. For example, the HLA region or genes that are relevant for cancer and many other traits. When we want to explore the effect of rare or novel genetic variants, it's very important to have deep phenotype data from any level of clinical care and as complete health records as possible in order to explore the effect of these genetic variants. Do they have any indications of disorders that could be associated with these rarely studied genes, for example? That makes our study much more powerful, that we are able to bring together different layers of data, both from the clinic and from different questionnaires and metabolomics or different omics data sets. So that gives a lot more strength to our study. I think the first thing other countries can say is definitely like, look at Estonia. If they can do it, why can't we? And I think what is very important is to have all the ethics and legal in place, especially being able to go back to people so that if you have any specific findings that you would like to explore, then you can contact these individuals and invite them into studies where you explore the effect of these genetic variants. If I had to give one piece of advice to another country that wants to start a biobank, it's to get everything legal and all the ethics in place before you start. And really think through the whole process, like what do I want to do, what permits do I need to have, what consent forms do I need to have, and then explore whether you want to have the possibility to go back to people and invite them into further studies based on findings, for example. I think it's really important to dream big, but then at some point do a reality check that we have this huge vision, we got funding for this, and we have the capacity to do maybe a slightly smaller part of it. In population scale research, you have to be ambitious. You have to set big goals and try to deliver something that's of benefit to as large a proportion of the population as possible. But there will be obstacles and there will be hurdles that we cannot overcome, either ethical or restrictions within the clinical setting, that we might not know enough about certain genes that we would like to use either in the clinic or in our research and return results to individuals based on different findings. But we might face different obstacles along the way that we cannot overcome, and that's where the practicality kicks in, that sometimes we have to give up some areas that we would like to explore or implement and just focus on what we can do. We were really happy when we heard about the Spark chemistry for the review, because as scientists we love innovation, and we also love it when we can do more within our budget. The new SPRQ chemistry gives us much more yield per smart cell. So this has allowed us to scale up our project and do more samples per week. And this will allow us to finish the project a lot ahead of deadline. For our biobank, the quality of the genetic data is really important because we want to be able to both use it for research and for clinical testing. So we really need high quality genomes and reliable variant calling so that we could use this information on an individual level as well. I think it's possible to focus on a specific disease area, or even pharmacogenetics is relevant for every individual patient. So if you have a strong phenotype, you can definitely manage with a much smaller cohort. In implementing genomics in healthcare, there are several low-hanging fruits that I think everyone should be working on. So pharmacogenetics definitely is one of them, because we will find something in everyone's genome that influences their drug response. This is definitely one of the first things that we need to start implementing in the clinic, and it can be done in any country, regardless of where you are and what level researcher you are. You can start with looking into different possibilities of pharmacogenetic testing, research and implementation. One piece of advice that I have is that we should all make an effort to implement pharmacogenetics in the clinic. I would definitely recommend to younger scientists to look into easily implementable genetic findings, such as pharmacogenetics or monogenic findings with high impact that are not too rare. This would definitely give them an easy place to start. Next, for the Estonian Biobank, we want to find funding to be able to sequence the entire biobank. And there are two avenues that we want to take. Based on these novel findings that we find in the genomes, we want to do research to find out what these rare genetic variants actually do. And on the other hand, whatever we already know about the genome that could be implemented in the clinic, this is something that we want to drive towards clinical implementation. I want to collaborate with many different countries in projects such as HiFi Solves and learn from each other internationally of how we could drive genomics into the clinic faster and better together. I think that there will be trailblazing breakthroughs on many different levels thanks to the technology that PacBio provides. It can be on transcriptomics, it can be on epigenetics, it can be in genomics and rare diseases and many different areas. The work that clinical geneticists have been doing with long reads and solving really tricky clinical cases using long read sequencing has really inspired me to look into the complicated regions of the genome. And it also makes me wonder, what are we actually missing in our research today? Starting over today would be really different from when we started 20 years ago, because the technology has advanced so much and long read sequencing has finally become affordable on a population scale. So, of course, if I started today, I would initially start with long read sequencing immediately.